the introduction. And first, I would like to thank my supervisor to bring us here because I'm not belong to the community. And I would like to talk about the mRNA display pipeline for protein biosensor constructions. And protein biosensors has applications in diagnostic and research, or as research tools. And in the past, many years, our lab has developed biosensors with different architectures. So one of them is quite generic, and it's kind of modular, and you can apply to different targets. So I show the scheme here. So it has 2 ligand binding domains, and one ligand binding domain fused to an enzymatic core, which the enzyme is inactivated by a calmodulin insertion. The other ligand binding domain is fused to a calmodulin binding peptide. So when the sample has ligand, it will bring the 2 components together and trigger the conformational change, activate the enzyme activity. And we can do colorimetric assay to see how much ligand in the sample. And ligand binding domains is a key element to determine the biosensor's specificity. So it mainly sourced from the PDB data bank. So for the one which do not have known ligand binding domains, AI turns to be a successful strategy at the moment, as my supervisor talked about yesterday. However, the success rate is not that high. Sometimes you still rely on selection system to narrow down the candidates. So it comes another goal of the project is to develop a high throughput selection system. So by looking at the literature, there are many display system has been developed for in vivo or in vitro selection. So mRNA display, which is my main workhorse, stands out because it has high diversity. It can display up to 10 to 13 molecules. So I would like to give an analogy. So the world population at the moment is 8 billion, so it's 1,000 times diversity than the world population here. And so the protein will be displayed on mRNA through a paramycin oligo, through a paramycin small molecule. And each mRNA will display 1 protein, and it forms covalent bond. So the bond is quite, the covalent bond can tolerate harsh selection conditions. And I think from 2020, we started to set up the mRNA display and optimize the platform, and we successfully finished each round of selection within 7 hours. So we test the enrichment factor is around 1,000 times. For each target, we normally do 4 to 6 rounds of selection. And the first library build is FN3-CON. It's a small protein, very stable. It has high expression yield. And we use this FN3-CON library build binders and then convert to sensors for many different proteins, such as liver toxicity biomarker proteins. And we also can develop conformation-specific binders against the protein small molecule complex. So I stop here. If you are interested, please come to visit my poster number 11.